Hi, my name is Thomas Funck. I'm a research scientist at Child Mind Institute. And today I'm going to be talking about an ongoing project, or the latest step in an ongoing project, about reconstructing 2D autoradiography sections into 3D. And today in particular, we're going to be talking about a technique we've developed to do morphology-based correction for batch effects in autoradiography, but also potentially in other modalities like histology. So for those who haven't seen earlier iterations of this talk, I'll quickly go through the data acquisition so we're all on the same page. Basically, we begin with a fresh brain. The brains have to be fresh because if you chemically fix them, then they can degrade the receptors that we're trying to measure. The brains are cut into 2 to 3 centimeter slabs, each of which are individually then shock frozen and then sectioned. The sections, which are effectively 2D, or almost practically 2D, are then bathed in a solution that contains a radioligand that will bind to the target receptors, and we can then digitize those to produce actual images of the receptor binding. Now the project is to take these 2D sections and put them into 3D, and to do that we developed a pipeline which does this quite effectively. The only problem is that once we had reconstructed the 3D images, we saw that there were batch effects that had been introduced. So I'll illustrate that in a second, but basically probably related to the incubation of the sections in the radioligand, there are mean shifts between different batches of tissue. And so you can see that here. So here we have a fully reconstructed image for the flumazenil ligand for the benzodiazepine receptor. And so you can clearly see that there are shifts in the average receptor density that we measure different pieces of tissue, in different pieces of tissue. So there's different approaches one could take to this. One would just be doing naive Z-scoring, but the problem with that is that if there is a real gradient along the rostrocaudal axis, you would get rid of it with the Z-scoring. Another approach would be to look at the border regions between the slabs of tissue and then try to do some kind of histogram matching or just matching the means or something like that. And that is indeed something that we initially tried. The problem, though, is that even though it produces a very uniform looking image locally, it still creates discrete jumps along adjacent pieces of cortex. And so that's something that we'd like to eliminate so that we can do all sorts of analyses like gradient analysis and other things. To that end, we developed a morphology-based batch correction. So this uses a cortical surface, like a mesh extracted from the T1 MRI of the donor. And so the idea is that on this mesh, we can, for adjacent slabs, find the closest pairs of vertices, and then calculate the average pair difference in intensities measured at those vertices, and then simply use those average differences in pixel intensity between the two slabs to then correct all of the slabs. So it's a very simple approach, but it seems to be fairly effective, as I'll hopefully convince you of in a second. So the idea is you'd maybe have your batches start, your tissue slabs on average, have this sort of distribution, and then at the end they have a corrected distribution that looks like something else. To maybe illustrate that for you a little bit, so here we have the vertices, the paired vertices in each of the adjacent bits of cortex. So here we have the rostral portion of slab 6 and the caudal portion of slab 5, and so the vertices with the same colors are the ones that are paired together. What this looks like in terms of measured intensities is what you see here. So we do have what look like very significant mean shifts between what should be nearby vertices. And so we can use this then to correct the slabs of tissue. And to see what this looks like, what the result looks like, is here we have again the uncorrected image, and then after batch correction, it looks much more continuous. There might still be some underlying batch effect, but it's at least much attenuated compared to what it was before. But so this is just a pretty picture. We'd like to put some numbers on this. How well does this really work? So to do that, we did a sort of theoretical validation using synthetic data. So we randomly generated vertex values with gradients along the medial lateral, rostrocaudal, and ventral dorsal axes. And we also included gaps between between vertices that actually have intensities to sort of mimic the sort of gaps we have in the actual data. So we generate 1,000 of these different synthetic data examples. And then for each of these, we introduce batch effects, random batch effects. And then, this is loading very slowly. And then we apply the batch correction algorithm, the morphology-based batch correction to each and every one of them, and measure the correlation between the original values and the corrected values. And so overall, this worked quite well. The average, the R squared, about half of the R squared values were between 0.95 and 1. And so overall, it worked very robustly. But of course, we'd like to see how this really works in practice. And the closest we could get to that was comparing the reconstructed autoradiography volume, the flumazenal volume, with a published flumazenal PET atlas, which comes from Norgard et al. In 2021. This is Gitte Knudsen's group. So there they took 16 subjects and with PET images for the flumazenal ligand, and then aggregated them together and smoothed them with a 5 millimeter full width maximum kernel. And so we did the same thing with the autoradiography volume, so they'd have the same sort of spatial resolution. And then with these two images, which in theory should match more or less, we used the Jülich Cytoarchitectonic Atlas to look at all the ROIs and then calculate the correlation between them. And so the result was pretty good. The R squared was 0.71, so it indicated a good correlation between the autoradiography, the batch-corrected autoradiography, and the PET atlas. Now, what is somewhat vexatious about this, however, is that before doing batch correction, the R squared value is actually 0.69. So you might say, okay, well, there's not much of an improvement in the batch correction using this. Why bother? But it's important to remember that the data are heavily smoothed in order to have them at a comparable resolution, and so the batch effects could be relatively small compared to the smoothing that we do. Also, the main reason to, or one of the big incentives to do this batch correction is so that we have smoothly changing receptor densities along the cortex. And that's important for all sorts of different analyses. And so it's good that we're getting closer to the PET atlas, but it's not the only factor that we have to take into account. And so that brings me to some future work that we've just started working on, and is using this, now that we have the batch effect corrected, we can start to look at gradients within the receptors, look at gradients between excitatory and inhibitory receptors, and then compare those across species between humans and macaque. But that is just, we're getting into that, and so that will be for next year, hopefully. Other updates on the project is that we've put this into a package called Brain Builder. So if anybody wants to do their own reconstruction, hopefully this provides an easy way to do that. The inputs are fairly straightforward and are just CSV files, so you can have your non-technical people organize the data and then send it to you and then run the pipeline, and hopefully it works well. So I'd like to thank many people who have supported this project and participated in various ways, including my current collaborators, Child Mind Institute, Ting Shu, and Mike Millam, my former PhD supervisors, Alex Teal, Alan Evans, and other great people at the MNI, Claude, PJ, Mona, and of course the people at Jülich Forschungszentrum, Katrin and Nicola, as well as Conrad Waxel, who has helped a lot throughout this whole project. So thank you very much for your time.
And Thomas, you're still there for Q&A, is that correct?
Yes, I am. Can you hear me?
Yes, okay.
Oh,
Yes, we're fine.
great.
Okay, so this talk is open for questions. And I'll start. There are probably several questions, but one is, you have a very nice method now for alignment. How exact is the section thickness? Is your thickness is supposed to be what, 10 microns or
20 microns?
20
microns.
Okay. Is every section 20 microns?
Yes.
How do you know? Because if
because the anatomist told me so.
I'm asking about
I'm relying.
Yeah. Oh, okay. Because sometimes the thickness is not really exact, and you can get slight variability. But that would probably be in your error bars. Okay, just to make your life miserable.
I guess it's hard to. That would be hard to measure after the fact. I mean, it's hard to take like a ruler and put it onto this.
This is fresh. Well, it's possible that you would be off by a few microns here and there. For example, different resistance of the brain when you have a lot of ventricle, when you have different orientation of the white matter. That it's a little bit of an aside, but I thought it would be interesting as your first question. Okay. Sorry.
Thank you very much.
Yes, additional questions?
If I could just answer Kathleen's question. The sections, the cryotome sections were set at 20 microns. So yes, as you meant, there's also difference in sectioning through the white matter and cortex and subcortical structures, give and take a couple of micrometers. That would only pertain to the histologically sectioned, histologically processed sections, so the ones process for the visualization of cell bodies myelin. There you would have these possible differences in thickness. The sections that were processed for receptor autoradiography, what we did there was incubate each section with a radio-labeled ligand which specifically binds to the receptor you want to analyze, and then we had to expose those radio-labeled sections against tritium-sensitive films. There you actually don't have a section thickness. You lose your thickness, right? So there we say section thickness is 20 micrometers, as opposed to saying it's 1 pixel, depth.
Yeah. Okay. Thank you very much. We have time, I think, one more question. Yes. Okay, so I think this will be brief. And is it easier to work with, you showed both axial and coronal sections. So obviously there's differences in size, but I would also think differences in texture. So is one easier to work with than the other plane?
So the axial sections that I showed are actually reconstructed sections.
Yeah,
so all of the
I was
sections
talking.
in the reconstructed data are coronal. However, we have multiple brains, and some of those brains, some of the tissue slabs have been sectioned dorsal, ventral. So once I reconstruct those, I can let you know next year.